comment box to ask questions or tweet at phyloseminar. Today's speaker is Wilder Wohns. Wohns. Sorry, you told me and I messed it up. We were joking about how it's not German, but then I used the German pronunciation. Wilder is a Rhodes Scholar and a PhD student in statistics at Oxford, supervised by Gil McVean, Yen Wang, and Jerome Kelleher. He did his bachelor's at Harvard in human evolutionary biology, where he worked with Terry Cappellini. He then moved to the UK to do his master's in Cambridge with Tuomas Kivisild, where he studied the effects of the Black Death using ancient DNA. Currently, he is developing methods to efficiently estimate the age of ancestors in a tree sequence and to infer genealogies with large numbers of ancient and modern genomes. Welcome, Wilder, and thanks for speaking to us today.
Well, thanks so much for that introduction, Eric. Thank you for having me. I'm really excited to be kicking off this series of 3 phylloseminars. And I'll start by talking about fundamentals of tree sequences. So to understand why, we should care about tree sequences, I want to start by talking about why we care about trees and why tree-based thinking has been so influential in evolutionary biology and for really since the time of Darwin. And I hope by the end of this talk you'll be on the same page as me and be a tree sequence evangelist and more familiar with the work we're doing and the general suite of software tools we've developed to manipulate and simulate tree sequences. So I want to start by showing you a tree that many of us have probably been looking at a lot over the past few weeks. So this is NextStrain from Trevor Bedford's group. And they take SARS-CoV-2 sequences and they run them through a pipeline which, among other things, builds a phylogenetic tree from the data and visualizes it beautifully here. So each tip in this tree that we're looking at is a SARS-CoV-2 sequence, and here it's colored by the country of origin. All right, and I think NextStrain is a realization of the power of phylodynamics and the power of trees more generally, because they're multiplying the usefulness of the coronavirus sequence data by inferring the geographic spread and prevalence of the pathogen, and this becomes a fantastic tool in the public health response to a pandemic. Here's another tree from NextStrain, and here we're looking not at just SARS-CoV-2 sequences, but actually SARS-CoV-2 in the context of related beta coronaviruses. So in blue, we have SARS-like coronaviruses, and in yellow, we have SARS coronaviruses. And if the previous tree told us something about the pandemic that's ongoing and allows us to track it in real time, this tree helps us understand the origins of the pandemic. So you can see, for instance, that the most closely related sequence to SARS-CoV-2 is this Yunnan bat sequence here in blue. And we'll come back to that in a few slides. And like all phylogenetic inference, the core data that builds this tree are mutations. And I'm hovering over one of the sequences here with my mouse, and you can see the nucleotide mutations that separate it from other sequences. And that's the core of phylogenetic inference because it helps elucidate the genealogy that generated this data. It's our window into how they're all related to one another. But if we think back to our introductory biology, we remember that alongside mutations, there's another fundamental genetic process that occurs, is recombination. All right, and does this tree take that into account? Well, you may ask, why should we care about that? And recombination is something that's really important to take into account in phylodynamics. So here's a paper also from Trevor Bedford and colleagues, 8 challenges in phylodynamic inference. And one of the challenges they point out is that many viruses are known to recombine at a high rate. And if you don't take that into account, you could be having errors in your inference. And so the way that viral recombination works generally is that if multiple viral strains co-infect the same cell, they could interact when they're replicating, and that would lead to viral progeny with genetic material from multiple parents. So that's how viral recombination basically works. And not only is it a general problem in phylodynamics, but specifically in coronaviruses, it's been known for a while that this is a pervasive issue. Coronaviruses recombine a lot, and SARS, the epidemic in the early 2000s, is thought to have originated in part through a recombination between strains in horseshoe bats. So viral recombination is something generally important to take into account, and specifically for coronaviruses, this is a pervasive feature. And obviously beyond viruses, tons of organisms of interest recombine. So this is a figure showing variable recombination rate in eukaryotes, but prokaryotes recombine too, and it's basically an inescapable fact in biology. Recombination is everywhere. So if recombination is everywhere, and at least in viruses, it's important to take into account in inference, what do we actually take into account? What's basically the effect of recombination for people interested in trees? Well, very basically, what recombination means in this setting is that different parts of the genome will have different histories. Okay, so this is the fundamental insight behind recombination and genealogies. Different parts of the genome have different histories. And to show what that means in terms of trees, let's go back to SARS-CoV-2. And let's look at this paper, which is hot off the preprint press as of last week. And in this paper, or this preprint, the authors were interested in understanding the evolutionary origins of SARS-CoV-2. So there's a lot going on in this figure, but let's just focus on 3 samples here: the human SARS-CoV-2 sequence, that Wohns sequence, which is a bat coronavirus strain, and then a sister lineage to that which infects pangolins. All right, so we're just looking at these 3, and these, the authors of this preprint wanted to know the evolutionary origins of SARS-CoV-2, and in part of their analysis, they wanted to see, was there evidence of recombination in SARS-CoV-2 or related beta coronaviruses? So they looked at one particular subregion of the coronavirus genome, these 4,000 base pairs that you see here, and they built trees along that subregion. So you can look at 3 of the trees here, and let's just focus for now on these 3 samples, and specifically on the human sample and the bat one. And you can see in these 3 trees, the bat and the human are most closely related. And that's what we saw in the tree from NextStrain as well. Overall, the bat sequence is the most closely related available non-human coronavirus to this SARS-CoV-2. But there's this loop region, this is the variable loop, and this is really important for the mechanism of this SARS-CoV-2 because it encodes binding specificity to ACE2, which is a transmembrane protein. So this region is really important for understanding how coronavirus invades human cells and basically is causing this pandemic that we're living through. And interestingly, at this variable loop region, human and pangolin are most closely related. So we have a different tree topology here. You can see that. There's been this switch of the green and orange. And that very simply is the result of recombination. And without going into too many details, the authors show that it's probably not recombination in the SARS-CoV-2 lineage, the human one, but rather in the green, the bat lineage. And through, this elucidates a complex phylogenetic history, and through a couple other arguments, they show that this strain had probably been circulating in bats for decades before it made the zoonotic jump that caused this pandemic. So an interesting conclusion, and a lot of it hinges on this understanding of recombination making multiple trees. Another example of recombination making multiple trees is incomplete lineage sorting. So here we're looking at results of the bonobo sequencing project. This was back in 2012, and we know that on a species level, bonobos and chimpanzees are more closely related to each other than either is to humans, right? So we can see that in this tree here, bonobo, chimpanzee, humans. These 2 share a common ancestor before either one shares a common ancestor with humans. But that's not true if you look across the genome and you compare all of them, the 3 species, to each other across their genomes. In fact, sometimes you see relationships that aren't consistent with that tree. For instance, chimpanzees and humans being more closely related to each other than either is to a bonobo. So how is that possible? Well, again, it's recombination making multiple trees. And this is a consequence of it. One more example about the effects of recombination are from this work by Terry Cappellini and colleagues in Nature Genetics. I worked with Terry as an undergrad. I wasn't specifically involved in this project. But in this paper, they found a really interesting and important enhancer to an interesting gene, GDF5. And the locus that they're interested in was associated with decreased stature in humans and increased osteoarthritis risk. So, they wanted to understand the evolutionary origins of this important region. And a great way of understanding evolution is trees, as we know. So they wanted to build a tree of that region, and they did that with 1,000 genomes individuals, so a diverse set of humans, some archaic humans, Neanderthals and Denisovans, and chimpanzee. And they built this tree, which you can see on the left here, and they found two main haplotypes. One of them was derived, specifically there was one derived allele they were looking at, and that was predominantly in non-Africans, but also the archaic individuals, and the other one was ancestral. And this was really powerful, intuitive way of understanding what happened to this locus, and it lended credence to their argument that the derived haplotype came from archaic individuals. So a great way of understanding what's happening there, and they built this tree at a 130 kb linkage block. But though you can identify a linkage block, if you're looking at that much sequence, there's always going to be samples that aren't consistent with the assumption of a single tree. And indeed, they found that a few hundred of their samples weren't consistent with that assumption, and they couldn't use them in the analysis. So while you get a great intuitive understanding of what happened to that locus with the tree, in reality, we know that there are multiple trees. So from these different modalities and these different subfields, we know that the current approach works quite well, and that's assuming a single tree. And that's brought us insight in phylogenetic, in understanding particular loci of interest, in understanding species-level relationships. But in the presence of recombination, there are going to be multiple trees. That's just reality. So a more complete model would take into account these multiple trees. We need more trees, right? But how many is more? So let's get a sense of what we're faced with here. Well, a good example of how many is more in terms of trees comes again from the 1,000 Genomes Project. So this is 2,500 diverse human individuals. We used our tree sequence inference algorithm, TSInfer, which you'll hear all about from Yan Wang next month. He's going to talk about tree sequence inference, so tune in on May 22nd. So we used this inference model, and we found that there were 529,000 unique tree topologies on chromosome 20. All right, so that's a lot of tree topologies, and that's just 1.2% of the human genome. So back of the envelope, that means there are 44 million trees across the human genome potentially. And each one of those trees has 5,000 tips because humans are diploid. So now if you want to represent all those and save them on your server, you have 44 million trees as Newark files, and that takes 5,800 gigabytes. Okay, but as a tree sequence, you can store that exact same information as only 13 gigabytes. So how is that possible? What is tree sequence, and why is it so much more concise than just having a bunch of trees? Well, let's talk about it. Let's get into the format and what a tree sequence is. So we understood why tree sequences exist. It's recombination, creating multiple tree topologies. Now let's figure out what that means. So let's have a little thought experiment. Let's say we have a population of interest. Let's say our population of interest is everybody in Seattle. So you sequence everybody in Seattle, you get all the genomes, and you put them in a hat. And I have this big old hat, you give it a shake. And we're going to draw 3 people from our Seattle genome hat. All right, here's our first person. And they're from West Seattle. And then we have our second person from Magnolia, and our third person from Tacoma, which is where I'm from. So now we have our 3 people, and we know humans are diploid, so we actually have 6 chromosomes. Okay, so now let's figure out how these Seattleites are related to each other genetically. Okay, and we're going to just use 50 base pairs of information. So what we're looking at here is our 6 chromosomes, and those are rows, and then columns are the 50 nucleotides that we're looking at. All right, and if you're eagle-eyed, you see that right away, there's only 4 sites here that are variant. So we don't need the other ones. We can toss them out. We don't need the invariants for now. And in red, we have the derived alleles, and in black, we have the ancestral alleles. So the original ones in black and the results of mutations in red. Okay, great. And also, we don't need the nucleotides. We can just code these as 1s and 0s. So 1 is derived and 0 is ancestral. And looking at this, many of you deal with this kind of thing a lot. This is what a VCF file stores. This is a genotype matrix. And this is encoded with complexity big O of m times n. So number of sites, 4 in this case, times the number of number of chromosomes, 6. Okay, so that's the storage complexity of a genotype matrix. All right, so we said we want to know what's the genealogy of our 6 Seattle chromosomes. How are they related to each other? What generated this pattern of variation? So I'm not going to talk about inference today. How you would infer that is going to be the subject of Jan's talk next month. But let's just say we have a crystal ball and we know what the gene tree at the first variable site is. And here's our gene tree. We have our 6 chromosomes. We see how they share common ancestors back in time, back to this common ancestor of all of them. And note that humans are diploid, and that means that the 2 chromosomes you draw from each person, they don't have to share a common ancestor with each other before they share a common ancestor with the others. That's what happened here. Right? A1 and A2 have a common ancestor before they do with the others, but B2 and C1 do not. Okay, so this is our gene tree. We looked in our crystal ball, and we got it. So let's put that above our genotype matrix and consider the first variant site. So at this first site, very simple, only A1 has the derived allele. So how is that reconciled here? Well, you just drop that mutation above A1, and A1 inherits it, and the others don't. And you're set. There you go. Site 1, site 0 sorted. Now the second site, how do you put that on there? Well, you have B1, B2, C1, C2. Okay, so how can you put one mutation on this tree to get that pattern of variation? Simple enough. There it is. The 4 of them inherit that mutation. Great. Now we could keep doing this all day, but let's make it a little more challenging and look at the third site, site number 2. Okay, how can you get B1, B2, C1 on that tree? Well, you can't. There's no way to get those 3 on the tree without a back mutation or recurrent mutation in there, something like that. With infinite sites, it's not going to work. So recombination must have occurred. There must be multiple trees, and it happens somewhere between 1 and 2. So we're going to represent recombination in the tree sequence as a subtree prune and regraft operation. So what's a subtree prune and regraft operation? It's exactly what it says. Let's copy the tree and prune a subtree from it. So there we go. We prune the subtree that leads to B1, and we regraft it onto the tree like so. Subtree prune and regraft. And then we'll do some bookkeeping. And look at that. We got another tree, and we put the mutation on there. And it all works out. Fantastic. That was a subtree prune and regraft. There was a recombination there in that ancestral history leading to B1 that led to that SPR, we call it, subtree prune and regraft.
So it's probably, it's probably worth emphasizing here that like the no back mutation thing here is really important.
Yes. You could, if you weren't assuming infinite sites, you could have back mutations on here, but for the purposes of this example, basically I'm just showing how you would need a recombination event to be consistent here. Yeah. So now we have our 2 trees, and the first tree explains those first 15 base pairs where we fit those two mutations on the tree, and then the second tree explains from 15 base pairs on, because like Eric said, we're assuming no back mutations, infinite sites. We have a new tree caused by recombination, and we put the mutation on there. So again, let's look at this 4th site. And assuming infinite sites, there's no way to get that on there. It fails the 4-gamete test. So let's copy the tree over. We'll do another subtree prune and regraft. Cut off B2, regraft it onto the tree, and there we go. So now we have 3 trees and 4 sites, and you can look at this and see that the tree sequence is a lossless representation of the genotype matrix. They're showing the exact same thing. You don't lose anything moving from the genotype matrix to the trees. Now, you might lose something moving back, but we can exactly represent the genotype matrix with this tree sequence. Okay, and we have 3 trees, and we see how much of the genome each one of those explains. 15 base pairs, 15, and then 20-ish for the last one. And we can get rid of our labels and forget which people each one of these nodes came from, and label nodes with integers. So note that because these are largely the same trees, we have the same samples, 0 to 5, 0 to 5, 0 to 5, and a lot of the internal nodes are the same too. 7 stayed the same for all 3 trees. 8's there for all 3 trees. 6 appeared with a recombination between these two, but now we have them all labeled. Okay, so that's the nodes in the tree sequence. The other fundamental thing is edges. So you'd be familiar with this if you work with graphs or trees generally. So how can we represent the edges here? Well, another way to represent them is through this notation, where we have the child node here. This is the child node of each edge, and then the parent that it leads to. So that up arrow pointing to 7, and the fact that it spans the whole genome here means that these edges exist for all 50 base pairs. So 0 to 7, there you go, 0 to 7, 0 to 7, 0 to 7. That edge exists for all 50 base pairs. And same with 1 to 7. So those are your sample edges, and here are some more sample edges. 2, but note that 2, the edge leading from 2 to 11 only exists for the first tree. Okay, and then there was a recombination event which affected that edge, and then it has a different parent, 2 to 6, 2 to 6, and then 2 to 8, 2 to 8. All right, so the sample edges, the sample's there for the whole tree sequence, but it can have different parents and different edge relationships. Here's the rest of the sample edges, and then let's look at the ancestral edges. So that leads from an ancestor to an older ancestor. And here it's worth making the point that these nodes, we know that these were our Seattle people that we picked in the beginning, but these are our ancestors. These are actual ancestors of these samples, right? These were people that lived at some time that gave some of their genetic material to the samples. And we can see that something interesting happens with ancestral nodes. They don't exist for the whole tree sequence. 6 only exists in this center tree for those 15 base pairs. All right, and what does that mean? Does that mean that 6 had some sort of small genome that was only 15 base pairs long? No, of course not. They had a whole genome, but through recombination, the only bits that survived to the present were the bits that exist in the tree sequence. Okay, so recombination has cut them up. And the other way of thinking about this is that the empty regions represent parts of the genome that don't make it to the samples. Okay, parts of the genome from those individuals. All right, so that's the intuition with the ancestral edges. You can see how they don't span the whole genome. And now let's look at some simulated data to understand what this looks like with something a little closer to reality. So this is an MS Prime.
We did get a question here. How are you deciding where subprepruner graft needs to occur on both ends of the operation?
It's a great question. And the answer is that we have a crystal ball here, and we just know. But if you're doing inference, that's a great question, because where you choose the inferred recombination event, where you think that happened, has major consequences for your inference. And Jan will talk all about that next month. But for now, we just say we know the whole history of these samples, and we know that the recombination happened at this position here in this ancestral lineage at time whatever. So
perfect, thanks.
No problem. So this is a coalescent simulation, 1000 kb, and we're looking at that same edge representation here. So we have the sample edges down here. These, as we saw, span the whole genome because we have all the material from those samples. And then we see the ancestral edges, and we've colored them by their frequency. And right away, you see that ancestral edges, they usually span, largest chunks of the genome. But as you go back in time, they get shorter and shorter on average. And that's, again, because recombination and making multiple trees is cutting up these haplotypes to make them smaller and smaller. And it makes sense, intuitively as well, because you're going to have more, directly inherited genetic material from your parent than you would from your ancestor 2,000 years ago. If you have anything from them, it's going to be some small bit of haplotype that's been chopped up over generations. Okay, so this fact that edges persist over multiple trees is cool and interesting for understanding population genetics and ancestry. But for the tree sequence, the really cool thing about this is it means that adjacent marginal trees are highly correlated, right? So these edges persisting mean that these trees from one tree to the next is essentially the same tree with the only differences caused by recombination. And to show that, let's go to some real data now. This is a different organism. This is Plasmodium vivax, pathogen that causes one version of malaria. It's from the P5X Genome Variation Project, and it was deconvoluted by Joe Zhu. And then I inferred ancestral alleles. You can ask me about it if you're interested, but basically we got all the pieces in place to infer a tree sequence from this. And we have a tree sequence of these 124 P5X samples color-coded by country of origin. And what we're looking at here is, in alternating shades of gray, the different marginal tree topologies of these 124 samples as we go along. So if we just start going along here, see we're moving this blue bar, and note the difference in each tree topology as we go along. They're largely the same, and the differences are SPRs, subtree pruner regraphs. Okay? And that, like I said, means that we're basically storing that same tree with minor changes as we go along. And that's why tree sequences are so concise. Okay, we'll see just how concise they are in a second. But first, let's understand how this is all encoded on the software side. And it's encoded as tables. So this is our example. Back to our original example, 3 trees, 4 sites, 6 samples. And we put mutations on this tree from our genotype matrix, but we can take them off too. You know, we have the tree. We can just say this mutation occurs above this node, 0 above 0, 1 above 11, 2 above 8. And you can satisfy yourself that you can move between those two and not lose anything. It's the same information. So that's what we call a mutation table, the way we store it there. The same with the edges. To store these edges, all we need actually is to put them, to note what's the child node and the parent node of each edge, what's the left and right coordinate of each edge, and then you get the same thing, an edge table, right? And that's the exact same thing as the edges. Then once you have those two things, you can take mutations off too and say, all right, these are all our nodes, or sorry, take all the nodes off. These are all the nodes. You could say when they occurred if you wanted as well. And then you just have a collection of tables, which exactly is how the tree sequence is represented under the hood in the software, right? And we said
that was such a great build sequence. Could you just go back and go through it one more time?
Sure, yeah.
Yeah.
Yeah, and Jan Wong gets credit for this magic move here, and but it's a nice way of seeing how you can take those off and put them in the table, right? So the mutation table, here's the edge table, and here's the node table, and there you go. There's a tree sequence. And the cool thing about that is you can see right away that if the genotype matrix was stored with complexity big O of m times n, the tree sequence is stored with complexity big O of m plus n plus r. So number of sites, there's the mutation table, number of nodes, node table, and then r, new recombinations making new nodes. That's big O order complexity. So that seems pretty significant for computer scientists. You might look at that and say, wow, that's a big saving. And here's what it looks like in terms of simulations. So this is storage performance. On the x-axis, we have sample sizes from 100 to 10 million individuals. So this is actually trees with 10 million tips. And on the y-axis, we're seeing what's the resulting file size, how big is it? And you can see, as you go along and you get out to 10 million individuals, you could store 10 million individuals variant data on a VCF, a compressed VCF, with 221 gigabytes. The compressed tree sequence, only 264 megabytes. So you're saving a lot. And if you look at this paper, in our paper here, we also show, if you scale this up to everyone alive, 7 billion people, how much would it take? And with a tree sequence, I think it was about 25 petabytes. Or sorry, with a VCF, it was about 25 petabytes. With a tree sequence, it was hundreds of gigabytes, I think. So that's what a tree sequence is. That's why it's concise. I want to show you now how you can use a tree sequence right now with Python or C. So if you go to this link, if you want to follow along and run this Jupyter Notebook yourself, you can go to my GitHub page in this repository, or you can just watch. I'll just take you through it now. So let's just get hands-on here and see how it works. So here's the notebook. You can run it yourself if you want. This first bit is just importing things we'll need, but I won't talk about it. And I hand created the example But let's just start here. So the documentation for everything is read the docs. TSKit read the docs. It's really, really well documented. And everything you need to know about tree sequences and the API is on there. And the way it works briefly is you just import the TSKit module, and then you load up this example I just created. All right, so this is our tree sequence example object. This is the top level tree sequence class. And let's see what we're dealing with. We have this draw SVG function. We call it draw SVG. We represent the SVG. And here we go. Here's our favorite example of the 3 Seattleites here. And okay, we want to know something about this tree. The easiest thing is how many trees are in the tree sequence? Well, there are 3. We just call num trees, looks it up in the object. There are 3 trees. How many nodes are there? Easy enough, 13. How many edges? 18. Remember that, it looks like there are more nodes, but a lot of these are just the same thing. We're only recording the differences. How many mutations are there? 4. Great. So we know all this stuff about the tree sequence right away. Now, let's say you're more interested in the individual tree topologies. You want to know something about those. Well, you take your tree sequence object and you call .trees. So if you're familiar with Python, this creates a generator over the trees, and you iterate over those, and you can look and say, all right, the first tree, 2 mutations, second tree, 1 mutation, third tree, 1 mutation, and that matches up to 1, 1. Great, so that's very simple manipulation of the tree sequence object. Here's that animation you liked again, Eric. We talked about what the tree sequence is stored as tables. Here we go. So it's just the same thing. We've got the mutation table, the edge table, and the node table. So what does that look like in the API? Well, you take your object, your tree sequence object, you look at tables. This is the collection of tables, and then you say, what's the node table? Here you go. Nice HTML rendered node table, where you can see all the IDs. There's 13 unique IDs or node IDs. Flags, is it a sample or not? So 1 is the sample. There are 6 samples. We can associate a population with each node. So if you're doing some sort of stratified simulation, or your data is stratified to populations and you want to keep track of that, you can keep track of that in TSKit. Or you have individuals. So we could have put the different Seattleites here as the individuals if we wanted. We didn't here. And then the time of each node. What time is it at? You need that to store the tree sequence. And then again, metadata. So, if you're working with some big data set, you can say this was, this bird from Costa Rica, and this one was from Honduras or something, or whatever population or organism you work with. You could store that in the metadata, which makes downstream stuff really easy. Here's the edge table. Again, we just need the left and right coordinate, the parent and child, and then metadata associated with that edge. So, maybe you're working in a setting where the edges are actually important as well, which could very well be the case. And then mutations table, IDs of the mutations. Which site does it occur at? So this actually kind of handles the infinite sites thing, because you want to allow multiple mutations at a particular site. There is a separate sites table, so that's where that's handled in answer to the earlier question, Eric. And then nodes, where does it appear? What node does it appear above in the tree sequence? What's the derived state? And this handles if there are mutations that are children of older mutations. So if it mutated one way and then mutated back or something, you can actually keep all of that in the tree sequence. You can keep that whole history. The violation of infinite sites, fine, no problem in TSKit with this kind of infrastructure. Okay, and then under the hood, something that people who program a lot in Python or do heavy loading or heavy duty statistical computation will appreciate is that under the hood, this is all NumPy arrays, right? So there's the nodes table, just the times of the nodes as the NumPy array. And I do a lot of work on inference with tree sequences, and the fact that these are all NumPy arrays under the hood means that you can do, like heavy lifting with the tree sequences in Python, utilizing NumPy, just directly from the object, which makes life really easy. But if you're a C person, you want to do some C programming, here's a snippet of what the C API looks like. It's documented at that same read the docs. This is a snippet of a Wright-Fisher simulator written in the C API. And there's a robust support for C as well. And under the hood, a lot of this is programmed in C. So that's TSKit briefly. And going back to the slides here, I'll just point out that this was all built by a community of a lot of people working hard on this stuff, and there's a lot of knowledge in this list of people contributing to it. We have people that are population geneticists, statisticians, they're into machine learning, they're biologists of all stripes, phylogeneticists, it's a diverse, and computer scientists, obviously. There's a lot of knowledge and a lot of collaboration. There's regular meetups, and there's a infrastructure set up. So if you want to get involved and tackle some existing issue, or you have an idea of something that you want to do on a tree sequence that you think the data structure is suited for, well, it's set up for you to join and get involved and talk with people on GitHub. So here's the link. Check it out if you're interested.
Wilder, I just want to double check something that you just said. It just really seems like there's absolutely nothing. There's no limitation about the sorts of mutations you can encode, in the sense that, although, I mean, in some of the inference steps, I think that you do assume no back mutation. Here, like, you could encode an arbitrary sequence of events that happen with, I think, arbitrary states. Like, you don't have to say, two alleles or whatever else.
No, yeah. So the data structure itself is flexible there. The inference, for inference, is a different story.
Right.
I
don't want to go there, but that's really neat. So everything is sort of fully general.
Exactly, it's fully generalizable. It can handle pretty much whatever you throw at it, and the whole idea is to losslessly represent variant data or genotype matrix sort of data. So it will handle that for sure. And you can go back and forth. Yeah. So let's talk about. So we've talked about what the format is. We've talked about why it's concise, how it all works. But now, if you're doing any sort of downstream analysis, or you want to use this in your work, you might wonder about statistics in tree sequences. All right. So if you're a phylogeneticist or a population geneticist, you might be wondering about this. So let's talk about this for a bit. And I have two examples here of statistics: the allele frequency spectrum, a fundamental population genetic statistic, and a statistic we defined for our tree sequence inference paper, genealogical nearest neighbors. So for phylogeneticists in the audience who don't deal with population genetics so much, you might be aware there's a lot of complicated formulae and metrics that population geneticists use to interpret population level processes. But I want to show you in this section that we can understand those processes with trees in an equivalent manner and possibly a simpler manner. So the citation for all of this is a preprint from Peter Ralph, Kevin Thornton, and Jerome Kelleher, which is a really great read. It's on bioRxiv, and thanks to Peter for his help with this section as well. So let's start by talking about the allele frequency spectrum. So if you're not familiar, the allele frequency spectrum very basically is saying how many samples are below a mutation in the tree. Okay, so if you go back to this original example we had, this is a slightly amended version of that. You have 6 samples, and you want to know for all 4, or now 3, slightly amended, 3 mutations in the tree, how many samples are below it. And we have possible buckets based on the fact we have 6 samples. We want to know how many mutations fall in each bucket. So with the tree, it's simple enough. You can just say this one has 1 mutation, boom, that bucket. This one has 4 samples, this bucket. This mutation has 3 samples, goes in that bucket. And that's basically the allele frequency spectrum. And you don't need trees to define this. You can do it, just with your variant data or your sequence data as well. And this is fundamental for a lot of population genetic statistics. But if you knew the true genealogy, so if you had the actual trees, the real tree sequence, real process that generated your data, you could use the branch lengths to give an equivalent definition of the AFS. So what does that look like? Well, here's a sequence of 3 trees, actually the exact same example. And what I've done here is I've color-coded the branches by the number of samples below each branch or each edge, the graph terminology we've been using. So blue, 1 sample below that edge, green, 2 samples below that edge. And if you, a basic principle of mutations is that they occur as a Poisson process on branches. So the sort of area that a mutation could fall on is the length of the branch, right? How long this branch is, multiplied by how much of the tree sequence it spans. So this branch, for instance, is this long, and it exists for 30 base pairs. So mutation could fall along this branch proportional to that length times that span. All right. So you can imagine an equivalent definition to the AFS, for instance, under infinite mutation rate, where that area, and borrowing terminology from the preprint, the area of the branch dictates dictates the allele frequency spectrum. And here I've filled in some arbitrary values that sort of visually match up with how much blue and how much green and pink there is in this example. Okay, so this is what you might say the expected AFS, and the actual realization of the AFS in terms of your mutations that you see is just where they happen to fall along those branches. So example of what they call in the preprint duality between the site-based versions of statistics and the branch length or the tree-based version of statistics. A really great example of that with real data is their figure 5 here. So this is chromosome 20 of 1,000 genomes again, and they're calculating pairwise differences between this is called the mean sequence diversity. So they take pairs of samples and they look in 1 megabase windows and they say for each pair, how many differences are you seeing between the samples? So at the top is just the classic way of calculating it with your variant data, doing it in 1 megabase windows, and these are of the different populations in 1,000 genomes. And in the middle is a branch or tree-based version of it. And this is done on trees inferred by Leo Spidel using Relate. So Leo will be talking about that on June 17th. So definitely tune in for that to hear about the really cool results and methods they're getting. So they took these trees and they found the tree-based version of mean sequence diversity, which I won't go into the details of, but it's analogous to that AFS version on sites and branches that I just talked about. So they calculate it on trees here, and then here's the ratio of the site to the branch version. And you note that there's some stochasticity, but generally it's a pretty good match. And that tells you that the tree inference that Leo did is robust. And the differences might say something about the effects of selection or mutation rate variation, this difference between the site-based version of the statistic and the branch-based version of the statistic. So that's what the AFS is on trees, but how do you actually calculate it? So the other part of this section, not only understanding these statistics, but also seeing that you can do them on tree sequences more efficiently, which is quite cool. And the way you do that is you assign a weight to each sample. So in this case, we just assign the same weight to each sample in our first tree. And then we propagate those values up the tree. Okay, we just collect them at each node, dynamic programming. And the way you collect them, if you collect them in different ways, you can calculate different statistics. But here we're just doing the AFS. So all we're doing is just adding them up. So let's see, how does that work? Well, there you go. This node had 2 below it, 2 samples below it, and we just sent them both up. Same over there. And then we send them up over on the corner there. There we go. That node had 3 samples below it, 4, and then the MRCA, the root node here, had 6. Okay, so now we've calculated our weights, and AFS is easy enough to calculate, or easy enough to find with that. You just say this mutation occurs above 1 sample, drop it in the correct bucket. Great. So if you move along, you'll see that this is calculated incrementally. And again, that's the key to the efficiency here. It's analogous to the reason that the tree sequence is concise. Things are stored as you go along. So this is a copy of that tree. We'll do our subtree pre and regraft it back onto the tree, do some bookkeeping, and there we go. There's our mutation. But we lost some of the weights, didn't we? So let's get them back. Send stuff up the tree, and there you go. So you can say, all right, that occurs above a node of frequency 4, you could say, and drop it in the correct bucket. And then your third tree, another SPR, but look, this time we only lost 2 nodes. So think about, put on your computer science hat and think about how efficient that is. We're saving a lot of this as we go along. There you go, the SPR, and then we propagate the values of the tree. and we put the mutation in the correct bucket. Okay, so that's an incremental way of calculating the allele frequency spectrum. And a side note, windows are something that people worry about a lot for how we're going to window our statistics along a genome. This is a natural window, right? Where did the recombinations occur? How much does each individual tree topology explain? But the output of this is something pretty striking, that tree statistics calculated on tree sequences, this one is Tajima's D, are much more efficient than using genotype matrices. So this is the TSKit way of doing Tajima's D on some simulated data. And this is some state-of-the-art methods on genotype matrices. So you're doing hundreds of thousands of variants per second, calculating the statistic versus hundreds of variants per second. So there are a bunch of statistics implemented in TSKit right now. Here's an incomplete list. There's also a general statistic function where you can take that way of assigning weights to samples and then propagating up the tree in however way you like, and you can define really quite a lot of population genetic statistics with that general function. So check out the documentation again to see how it's done. But the take-home point here is that once you have your tree sequence, you don't need to go around to some other program and install it or change the format, do anything like that. You just have to do .fst or .Tajima's D, and then you get the statistic. So one other statistic I'll talk about briefly is genealogical nearest neighbors. We defined this in the tree sequence inference paper, and this is a topology-based metric. And it's just a cool illustration of the sort of thing you could do with a tree sequence. So let's say we're interested in this node here. We like this node. We want to know who is it related to genealogically among our other samples. Well, there's a lot of ways to do this. A lot of ways are based on shared alleles or inferred shared haplotypes. Huge literature on that sort of thing. But what if you had the tree? Well, one simple way of saying how they're related is just look up the tree, like literally up the tree, and who's the parent of that node? All right, here's parent of this node, and then who are the other children? Well, it's these, and these are the nearest neighbors. And that's all there is to it. It's a very tree-based way of thinking about relationships. And you can see in real data that it actually brings out really fine-scale and complicated population-level relationships. So this is schematic of the 1,000 genomes. We built a tree sequence from 1,000 genomes. Here are the populations or region-level populations of 1,000 genomes. And let's say we are interested in HG0 1933, a Peruvian admixed American individual in 1,000 genomes. And here we go. Here's one of their haplotypes. Who are the nearest neighbors of this haplotype? Well, it's these 4 individuals, right? We went up the tree and we looked at the other children, these 4 individuals, and you can see they're from East Asian populations. Well, let's look along the tree sequence and do that at every tree topology. This is what you get for this individual, one of these individuals' chromosomes. At this region, the GNN fraction, the genealogical nearest neighbors fraction on the y-axis, and the position on the x-axis. At this position, all of the nearest neighbors are East Asian. Okay, and over here, they're from sub-Saharan Africa. Over here, they look like they're European, possibly admixed American. And you get right away this intuitive understanding of the ancestry of this individual, right? It looks like they probably had a parent or grandparent, one that was of East Asian descent, one of sub-Saharan African descent, and one of European or admixed European descent. And that's without any concept of time. It's just the length of the haplotype shows that they haven't been broken up enough, so that they're probably just a few generations removed. So if you look at the other haplotype of that individual, you can see that's what it looks like. Totally different history. Like we said before, there's no reason that they should have any similarity. One's paternal, one's maternal. And you can do that for all Peruvians. So there are these 2 haplotypes for that individual. Here are all Peruvians. So now what we're looking at is, again, the GNN fraction on the y-axis, but on the x-axis, the haplotypes. So these are, I think 130 Peruvians, or 260 because they're diploid, 260 chromosomes of Peruvians and 1,000 genomes, sorted by decreasing European ancestry. And we've highlighted this first chromosome we looked at, and then the second one. So it's sort of like integrating over this and then stacking all the ancestry from different regions.
That might have been an answer to a question that just popped up, which is what exactly do you mean by a marginal tree?
So marginal tree means if you had gone back to, I'll just zoom back to, there's a schematic tree sequence we've been working with. A marginal tree is just like, you just look at this tree, the marginal tree topology in the, if this is the tree sequence, this is one marginal tree, this is another marginal tree, this is another marginal tree. So that's what that means. We can send those back up again, put that back. So that's a great question. That's what that means. So, there's, like we said, there were, I think, was it 500,000 or something like that, tree topologies on chromosome 20. So this is there's 500,000 trees calculating the GNN. And then this is integrating over that and summing the ancestry from different regions for all the Peruvians in 1,000 genomes. Okay. And then, if going from here to here was a integration, then we'll just integrate again and look at all the populations in 1,000 genomes and relate them to one another. So on the x-axis, we have a heat map. On both axes, we have populations in 1,000 genomes, and we're seeing who are the nearest neighbors of individuals in that population. So let's find our Peruvians. Here they are, PEL. Okay, and we see they have the most nearest neighbors among other Peruvians, which makes sense. But after that, it's Mexican and then Colombian. All right, and you can see there's this admixed American grouping here. There's this lightly colored block showing the geographic affinities to one another, and then a larger grouping with Europeans, because there's a lot of European admixture among those populations. And you can see, among South Asians, among East Asians, among Sub-Saharan African groups, this sort of broad level genealogical relatedness, right? And this is all at the root of it, just built from marginal trees, looking at the nearest neighbors, and then various steps of integration. Here's the same thing with the Simon's Genome Diversity Project. So that's many more populations, and you can see, from more fine scale relationships there. But the sort of coup de grace here is the UK Biobank. So those two examples, 1,000 genomes and Simon's Genome Diversity Project, are hundreds or a few thousand samples. This is almost 500,000 samples, right? And again, humans are diploid. That means that there's 1 million chromosomes that we're considering here. So we built a tree sequence from 1 million tips. So that's, imagine like hundreds of thousands of trees with 1 million tips stored in a tree sequence that you can, that could fit on a laptop easily, just a few gigabytes. And then we do GNN on that massive tree sequence of UK Biobank. And we can see the heat map of relationships of people born in different parts of the UK to one another. And you get these really amazing fine-scaled relationships just from this simple genealogical nearest neighbors statistic. So you can see, for instance, people born in Cumbria, right? One region of the UK in northern England. Who is their nearest neighbors? Well, again, people from Cumbria. But then you can see this relationship to other other groups of northern England. You can see Scotland and Northern Ireland have a closer genealogical relationship. Wales has very tight, nearest neighborness with other Welsh individuals. The southeast, this part of England has had more historic migrations, and you can see broader scale affinities. And you can drill down even more and say, UK Biobank has rich information not only about phenotypes, which is a whole different story, but about where people are from. And you can say, all right, where were you actually born? Where are you from? City of Edinburgh. Well, who are your nearest neighbors in Scotland? Or you can do the same thing for people from Yorkshire or from Camden and City of London. So, a great illustration entirely from the trees of how you can see how very, very large numbers of samples are related to each other. So I talked for a while there, but I'll just briefly show you what statistics look like in TSKit. So we'll go back to the notebook and back to our example. So let's just draw our example again. Here we go. These are our favorite, 3 trees. And, let's say you've figured out how to get your data into a tree sequence, and now you want to do all these downstream analyses. Like I said before, easy as a keystroke. You just do allele frequency spectrum. You can pass these different parameters, which I won't talk about now, but you can figure out however exactly you want to calculate your statistic. Pairwise diversity we talked about earlier, calculate that efficiently. And there's this full list, or you can implement your own. So once you have it as a tree sequence, you can do everything you need downstream, or a lot of things you need downstream with it. For the phylogeneticists in the audience, I thought I'd talk about parsimony briefly and show you a very simple version of Sankov algorithm. So we have a tree here, 6 tips, and let's say we're trying to reconcile some observations with this tree and say what's the fewest number of transitions that explains the fact that we have a red allele, a blue allele, and a green allele, and then these observed genotypes. Okay, and interest of time, I won't go into too much detail, but basically you can run this map mutations, which is reconcile your observations with the tree. What's the fewest number of transitions that's consistent with this tree? And you can see that TSKit tells you that the ancestral state was probably that red allele, and then there's a mutation above node 4 to cause the blue allele, and then a mutation above node 5 to cause the green allele. So some simple parsimony implemented in TSKit as well. And then here's genealogical nearest neighbors. You just have to define your reference sets. So we defined our reference sets, and then you can just run GNN and see how each chromosome is related to your 3 reference sets. So very brief on the statistics, but I want to get into the simulations in the last few minutes here. So there are 3 simulators, or 2 simulators, and then a standardized demographic aware simulation sort of framework that have been written and are available. So let's go back to the notebook again, and we'll talk about MS Prime. So MS Prime is a really widely used simulator now. It's an efficient re-implementation of the classic MS simulator, and the documentation again is MS Prime read the docs. But once you install it and import it, you can just basically run MS Prime simulate, specify your sample size, your affected population size, and then out you get a tree sequence. And this object is just like the one we worked with before. You can do everything we did before on this, run your statistics, check it out, go through the trees, all that kind of thing. So let's just look at the first tree that we simulated, right? And this is what it looks like. We just ran MS Prime to create that. So let's say you're interested in the branch lengths on this, on your simulated tree. Well, you can say above node 7 on this tree, here's node 7, what's the branch length? It's this. And then what's the total branch length? Here you go, first tree, total branch length. So easily look at the tree that way. Or you want to move around the tree, you're doing some sort of tree-based operation and you want to have a pre-order traversal, you can traverse it in different ways, standard tree or graph traversals. So I started by talking a lot about recombination. So naturally, you want to have recombination in your simulator as well, and MS Prime handles that easily with just recombination rate. Set your recombination rate, set the length of sequence you're simulating, and when we rerun that simulator with some recombination, we get 3 trees. You can see how much of the 10,000 base pairs we simulated do these trees span. Mutations. You obviously want mutations on your trees too, so you just have to put in a mutation rate. And then now we have a tree that we just simulated here with 9 mutations. Okay, so the real, one of the really cool things about MS Prime is, again, efficiency. So that's a theme today, tree sequences being efficient. So let's simulate 100,000 10 megabase chromosomes. All right, so we're going to make a tree sequence of 100,000 10 megabase chromosomes. I just ran it, and see how long it takes. So in 5 seconds, we created 45,000 trees, each one with 100,000 tips. Okay, so we got a lot of data in a very short amount of time there. Not only was it quick, it's only 18 megabytes that we just created. So we can load that up, 38 milliseconds. We can iterate over all the trees, 187 milliseconds. And we didn't add any mutations in that original one, but now let's add some mutations. And we know that at least under the coalescent, the process of, with neutral mutations, process of creating the trees and the mutations are separate, so we can add the mutations on afterwards. So we're adding some neutral mutations on 2 orders of magnitude more, higher rate than in humans. So in 16 seconds, we just created almost 5 million variants. All right, so now we have this tree sequence of 100,000 tips and tens of thousands of trees, 5 million variants is 194 megabytes as a tree sequence. If you did a bunch of Newick files, that'd be 191 gigabytes. Or if you made that a genotype matrix, 453 gigabytes. So much, much more concise as a tree sequence than Newix or the genotype matrix. So that's an intro to MS Prime. I'll just briefly mention SLIM, forward time simulator, if you want to not do coalescent simulations, but you want forward time simulations. SLIM is a great tool for that. There's command line interface, graphical user environment. This is really cool. Slim GUI for Mac, so you can watch your populations evolve and change, and you can have splits and whatever else you want in real time with this really nice interface. There's standard PopSim, which is new as of just a few months ago. So if you're interested in any of these organisms, you work on these organisms, in one centralized place, you could get the chromosomes from those individuals, the recombination maps, published models of evolution in those individuals, in those organisms. So for instance, for humans, you can look at published models of the Out of Africa event, or ancient Eurasia or anything like that. It's built right in. You don't need to go to the supplement of the paper and dig out all the parameters. The parameters are in here in the model, and then you can do a forward or reverse time simulator and then get your tree sequence object out. So to sum up all of that, I hope I talked to you a bit, convinced you a bit about why you should think beyond a single tree, why that matters, and actually in pretty much every situation of organism you'd be interested in, recombination rears its head and makes multiple trees. And there's now, a really efficient solution to that, which is tree sequences. So fear not, there's a format that will handle it concisely. You can interact with it easily with Python, with C. You can calculate statistics on it. You can simulate forwards and backwards with it. And you can see how that comes together from all sorts of different modalities of computer science, biology, to make this sort of unified framework for understanding organisms that reproduce with recombination. So next month, you'll get to hear all about the inference side, which I know people are very interested in. You'll hear from Jan about how do you infer a tree sequence? How do you go from the variant data or the sequence data to the sequence of trees? And then once you have the tree sequence, how do you date all your ancestors? How do you know what the branch lengths are? Because we know the branch lengths are really important for a lot of the statistics we're interested in. And I'll just briefly mention what I'm interested in, which is inferring these tree sequences from ancient and modern samples. So there's tons of ancient samples available now. How do we leverage that in an intelligent way to produce tree sequences that maybe look a little bit like this, where you have dangling nodes or you dug up someone's ancestor? And then how do you understand those relationships over time? And how do you think about classic population genetics in terms of tree sequences now? So with that, I'll just leave you with some citations for what I talked about, and then say thank you to Jan, Jerome, Peter, Gil for his guidance, Ben and Daniel, and then all the TSKit developers. And yes, thanks very much.
Thank you, Wilder. That was awesome. Yeah, really pretty stunning work there. So let's see. I just wanted to start by thanking the people who have asked questions. Let's see. I'm actually losing. Let's see. So Dylan Taylor asked the question about subtree prune regraph needs to occur. And let's see. Gregor Gorjank asked about what you mean by a marginal tree. So thanks for those questions. Keep on posting. So I'll ask some other questions that showed up. So can the generalized nearest neighbors cope with a more distantly related population? So if we wanted to use the kit to compare young but closely related species that might be hybridizing a lot?
So yeah, that's a great question. It's so far been used in sort of within species settings. But in theory, it could work with sort of species tree level hybridization and that sort of thing. How that in practice plays out, I don't know, but it's a great question. And in theory, there's no reason that it shouldn't. It's just a general principle of how samples are related in trees.
When I started reading that question, I thought it was going to lead to more distantly related than just the nearest neighbors. But I guess that wouldn't be too hard to do. I mean, you're looking at the sister,
Yeah.
go up 2 nodes, and then look at the ensemble of all the people,
Sure.
or individuals. Anyway.
Yeah, exactly.
Jan wanted to comment that Forward PP is a C simulation library that outputs tree sequences. Noah Rose asked, with respect to efficiently calculating existing PopGen statistics, to what extent do you feel newer tree-naive statistics will better capture the signatures of selection or demography in question?
That's a great question, and I think probably the best placed person to answer that will be Leo in his talk, because he's been doing some work on selection with trees and comparing that to the signal selection you get from classic frequency-based or haplotype-based methods. But it definitely seems from his work that there is a lot to be gained from considering, the inferred trees and signals of selection trees. So obviously, if you knew the true trees, signals of selection would be apparent in basically, as Leo puts it, lineages outcompeting other lineages and rising the frequency that way. So if you sort of riff on that theme and say, all right, we have an approximation to the truth, you should be able to get something more than just looking at, allele frequencies and that sort of thing.
Right. Yeah.
I'll leave it to Leo to really answer that.
Yeah. Speaking of statistics, let's see. Is it sort of known what the ensemble of sort of PopGen statistics are that you can define as a TSKit statistic? Right. I mean, I don't know. I mean, are there any limitations? Are there things like statistics that you would want to compute that you can't compute using this representation? I mean, I guess the stupid answer is that you could always, take the tree and turn it back into a flat matrix.
Yeah. So in that sense, no, right? Because if it's a lossless representation, there's nothing you couldn't do with a tree. Now, I can't think of off the top of my head any examples where you actually would go back to a matrix unit calculation. So I'd have to say off the top of my head, no, I don't know the definitive answer to that. But I know that, for instance, a lot of population genetic statistics can be thought of basically as variations on the AFS, or if not, there's some derivation of it with, multiple different paths from the AFS. But yeah, I don't know the definitive answer to that. But in theory, no. Yeah.
Cool. Peter commented to Noah's question, adding on to what you said. We don't know yet. We need to do some tests. It could depend a lot on how well TS inference works on practice. I think this is the multi-species thing. There was a bit of a discussion about whether the tree transitions have to be an SPR, and Jan says no. In fact, in inference, it often isn't. It's often several SPRs. Let's see. And I guess is there any proof that actually this is sort of the most compact way in which you could represent?
Well, I mean, there are other compact methods. Positional Burrows-Wheeler transform is quite compact, and that's mentioned in the tree sequence inference paper. I don't know beyond that about more compact ways of representing the data, but this kind of goes to there's a quote from Joe Fosenstein I like a lot, which is to understand, the data, you think about the process that generated it. So, maybe there are more compact ways of compressing it, but this is nice because it's compressible, but it also makes sense, right? That's what generated the data. So.
And beyond making sense, you can obviously do all sorts of interesting queries of it, where it's like, if it was just like a big gzip or something stupid, you're like, well. Let's see. Julia asked, what are some of the challenges of integrating historical samples into tree sequences?
Yeah, well, that's a great question. There's practical challenges about just the nature of ancient data. being, broken up and fragmentary. But if you have, so I think the way that I've been thinking about it is separating it into the sort of lower coverage or genotyped ancient data, where you're really dealing with more of a missing data problem in addition to the sort of chronological problem. And then the really high quality stuff, so like a Neanderthal sequence, which is deeply sequenced, and you're not really dealing with missing data, but rather with just an old sample. So that's kind of the way I think about it. Is in one case, building it into a tree is quite hard just from the missing data perspective, but in the other case, you basically have a sample that you can just treat like a sample that coalesces at at or older than its age, right? So that gives you the lower bound on its coalescence as you radiocarbon date, and then otherwise you just treat it like any other sample. But yeah, for the stuff that's lower quality, it's there's a lot of information in there as well, but it's just handled a little differently.
Cool.
Mm-hmm.
We got one question from Twitter. Are some of the large data sets available as tree sequence downloads, such as 1,000 genomes?
Yes, yeah, and there's a Zenodo link. So if you go to the supplementary information of the Nature Genetics paper, the tree sequence inference paper, and you basically search Zenodo, you'll see downloads for 1,000 genomes and Simon's Genome University project. For biobanks, since obviously that's protected, you can't download the sequence information, but the methodology is there if you want to do it yourself.
Cool. Patricia Brito asked a question about phasing genomes before analysis, but I think that's probably gonna get that's maybe for next time.
Yeah.
I said Julia before that's Julie Kreiner. I think we should wrap up here. That was a great seminar. Thank you very much for sort of kicking things off, and I think everybody is pretty excited to hear about the next few talks.
Yeah, definitely. Well, thanks so much for hosting it. Really appreciate it.
All right. Well, see you next month.
Okay. See ya.